Hi everyone. This video is about chemiosmotic hypothesis. This is a very important topic in the chapter of photosynthesis in higher plan. So let's start the discussion. See, the main idea of the theory is that it explains us how ATP synthesis happens in the chloroplast. The same theory we can apply for mitochondria also, how ATP synthesis happen during aerobic respiration in the inner mitochondrial membrane. But here I'm specifically discussing it for the chloroplast, how ATP is synthesized during photosynthesis. So we need to recall here a few points about photosynthesis, that there will be two set of reactions. The first set of reactions are known as light reactions, and second set of reactions are known as dark reactions. Now, in the light reactions, there is formation of some energy intermediates such as ATP and NADPH, whereas these are utilized in the dark reactions for synthesis of sugars. Okay, so we will be seeing how this ATP part is synthesized in the light reactions. Now, moving to the next part. So as per the theory, ATP synthesis is linked here to the development of proton gradient across a membrane, which is specifically the thylakoid membrane here. Okay, so this thing we need to understand properly, that in the explanation we will see that how a proton gradient is developed across this thylakoid membrane. Now, if you remember, the thylakoids are coin-like structures, just like this. They have a coin-like shape with this membrane. This is thylakoid membrane. And inside there is a lumen. Okay, so we will understand here that during the process of light reaction, we have this process known cyclic photophosphorylation. Okay, so there will be an electron transport across various carriers, and that process will lead to the generation of protons, which are going to accumulate here into the lumen. And because of this, what happened? We see the gradient formation because stroma part is going to be deficient in the protons. Okay, whereas lumen will have excess of protons. Now, when this gradient is broken down and the movement of protons happen from lumen to the stroma, that process leads to ATP synthesis. Okay, so the most important thing to understand is why this proton gradient is generated. Like, what are the various events which lead to accumulation of protons into the lumen and deficiency of protons into the stroma. So these events have to be understood. So just look at the diagram here. This pink color, dark pink color, is showing the thylakoid membrane. Okay. Then inside we have this lumen. It is written that it has high concentration of protons. And this is a stroma outside. The whole fluid matrix will be the stroma, which has low concentration of proton. So basically we have to discuss this thing only, how this low and high proton concentration, or you can say this proton gradient, is getting generated. So to understand this, we need to see various structures which are represented on the membrane. We have this photosystem 2, P680, then this photosystem 1, P700. And in between these two, you can see cytochrome B6F, which is a part of electron transport chain. And then we have this ATP synthase also. Okay. And then you can see some mobile carriers. One is the plastoquinone. You can represent it as PQ. And second, we have plastocyanin represented as PC. Okay. Now let's understand how the electron transport will happen during non-cyclic photophosphorylation and how the protons are getting accumulated into the lumen. Okay. And how they are getting deficient in the stroma. So we'll start from photosystem 2. Okay. Now the location of photosystem 2 is, it is located towards the inner side of thylakoid membrane. Okay. Now when light energy falls over it in the form of photons, so the electrons from the reaction center gets excited, and those electrons, they are accepted by the primary electron acceptor, which is pheophytin. Primary electron acceptor. Now this pheophytin will be transferring it to the next carrier, which is plastoquinone. Now about the nature of plastoquinone, it is primarily a proton carrier. So while transporting the electron, it is going to accept a few protons from the stroma. So this PQ will be reduced to PQH2. Now electrons are taken by cytochrome B6F. Where does the protons go? They will come to the lumen. Okay, so this is one of the reason why protons are coming into lumen. Now here only you will see the second point also, that when this photosystem 2 loses its electron to pheophytin and plastoquinone, it becomes unstable. And to stabilize this, we have the ultimate electron donor, water here. So water is going to be oxidized by this light, and this photooxidation of water gives us oxygen, protons, and electrons. Electrons go to the photosystem 2 to stabilize it, and proton will be coming into the lumen. Okay, and oxygen will come out from the stomata. So this was the second reason why protons are getting accumulated into lumen. Now, moving further from cytochrome B6F, the protons will go to plastocyanin, which take it to photosystem 1. Now, photosystem 1 gets excited and the electron move to ferredoxin. From ferredoxin, what happened? We have this enzyme FNR. FNR stands for ferredoxin NADP reductase. So this enzyme complex is situated on the outer side of the membrane, and while reducing NADP plus to NADPH, it is going to utilize a few protons from the stroma because it is on the outer side of the membrane. So again, this process is although not accumulating the protons to the lumen, but this is using the proton from stroma. So again, increasing this gradient. So in total, we have these three reasons. One, photooxidation of water. Second, this plastoquinone. And third, this FNR enzyme. So these three collectively will be increasing this gradient of the protons, where lumen is rich in proton and stroma is deficient in protons. Now to break this gradient, what happened? We have this ATP synthase when protons are trying to come out into the stroma. What we see is that in this ATP synthase, we have two parts. One is this CF0, which is an integral protein. It carries out facilitated diffusion of the protons. And then we have the second part, CF1. See, C indicates here the chloroplast region. We have a similar enzyme in the mitochondria also, which is represented as F0 and F1. Okay, so this CF1 is a peripheral protein which is protruding outside into the stroma. And what happened when these protons are getting facilitated outside towards the stroma? This brings out some conformational change in the CF1 part, and that leads to ATP synthesis. So you can see this phosphorylation happening. This is known as photophosphorylation because it is happening with the help of light. Okay, so this was the whole process of ATP synthesis in chloroplast. Now let us summarize all the point. So first point is the photolysis of water happening on the inner side of the membrane. As I said, the photosystem 2 is located towards the inner side. So that's why it will happen on inner side. So whatever protons get generated, they are going to accumulate into the lumen. Now second point, the transport of protons from photosystem 2 towards pheophytin and then to plastoquinone and to cytochrome B6F and further. So here plastoquinone is a proton carrier. So that's why while transporting the electron, it will be removing some protons from stroma. And third point, due to NADP reductase enzyme, which is present toward the outer side of the membrane. So for NADP reduction, it will be removing some protons from stroma. Okay, so we can say due to all these reasons, there will be a proton gradient across thylakoid membrane, as well as there is a measurable decrease in the pH. It's quite obvious because protons are getting accumulated, so that creates an acidic environment and pH reduces. Okay. So this is a bit of explanation about ATP synthase. ATP synthase has two parts, CF0 and CF1. CF0 is an integral protein which is embedded in the thylakoid membrane, and it carries out facilitated diffusion of protons. CF1 is a peripheral protein which is protruding on the outer surface towards the stroma side. So the breakdown of gradient will provide enough energy to cause a conformational change in CF1, which leads to production of ATP. Okay, so we will summarize the whole process in nutshell. So we can say the chemiosmosis require four things. a membrane, which is thylakoid membrane here. Then we need a proton pump. We need a proton gradient here, and we need ATP synthase enzyme. So that was all about chemiosmotic hypothesis. Thank you very much. I hope this video helped you to understand this topic very well.